So it's a great privilege then to welcome back this afternoon Professor John Bryant, who is the Emeritus Professor of Cell Biology from Exeter University, and who's been there, as we heard this morning, 22 and a half years, I haven't forgotten, and who's now doing a number of other things. One is to write and to lecture extensively in the field of bioethics. And John, together with another John Sell, has written this excellent book, Life in Our Hands, which I hope many of you here will be able to purchase from the bookstore. And John has also been involved in setting up bioethics course, not only in his own university, but also helping others in different universities to do that. And this afternoon he's going to talk to us now about stem cells. So thank you very much, John.
Yeah, thank you, Dennis, very much for the kind introduction. As I said this morning, my own research has mostly been on the replication of the genetic material, DNA, in plants, the process of copy prior to cell division. If you want to really know what to sort of buffer now, I might tell people that I've been focusing over a period of 35 years on 2 minutes of a living cell's life. So that's sort of fairly boffin-like. And it's a very vital 2 minutes. It's the 2 minutes in which a cell prepares to replicate its DNA and then starts doing it. And in plants, that happens in the adult plant in stem cells. Adult plants maintain populations of stem cells which contribute to the growth of the individual organs. And so the root and the leaf and the stem and the flower and the stem apex all have populations of stem cells. So I've been working with plant stem cells. Unlike animal stem cells, they're very easy to culture in the lab and they will revert to being totipotent very readily. Totipotent means they're capable of doing anything, which means that we can generate plants from stem cell populations. So that's the kind of my background to which I come intellectually into this debate about mammalian stem cells and in particular human stem cells. So that's the first introductory comment I want to make. And the second one is that if we're trying to think about how as a Christian or a religious community we're going to bring our ethical thinking to bear, we need to understand that the holy writings of Judaism, of Christianity, and of Islam say nothing about these topics. And indeed, we need to understand that those holy writings stem from a period in which human pregnancy was not understood in the way that we now understand it. Indeed, the human egg was not discovered until the 18th century. And this is why in the biblical writing about childbirth and pregnancy, a woman who can't have a child is usually referred to as barren, as if she was ground that was infertile for the reception of the seed that the man provided. And indeed, if you go back to the Middle Ages, in Europe, you'll see pictures of sperm where the whole little human was embedded in the sperm head, the homunculus, the little man. On the grounds that all the woman did was provide the ground. And that is the understanding of conception that we need to keep in the back of our minds as we discuss these issues. And I'm sure Garrett will unpack this in very much more detail. And it may affect the way we think about these issues. It may not, but it may do. Now, before we talk about stem cells in general, I want to talk about the world in which we're debating this issue, because it's been an issue that has been very much affected by the mores of our time. We live in a political world. One of Bob Dylan's less-known albums, this is one of the better songs on that album, so it's one of his less-known ones. I have them all, so I don't know them. But it's very apparent, and he sings about a culture in which decisions are made for political expediency without any underlying love. Is what he's talking about. We might say agape or charity in the context in which we're speaking. But we're also living in Britain anyway, in a world which is aggressively secular. You can't say that. So much so that the practicing Christians have to, in some way, justify that they are actually sane. When in a society in which Richard Dawkins can get on the television to talk about anything, whereas Nicky Gumbel is not even heard of by the media, as an example, as someone who's very active in Christian evangelism, and a thoughtful lawyer to boot. And then we've got postmodernism, which mixed with the modernity of science and technology has infiltrated, particularly into the media, this idea that there are no meta-narratives, no objective truths, and if you actually hold that there is a God, for example, that is a meta-narrative that you can't possibly hold of that. Or if you do, it's okay if I have my own version of that. And again, another quote from Bob Dylan, "You're right from Russell, I'm right from mine," perfect description of postmodern ethics. So let's go back to these wonderful pictures which were published in the Biochemist in 1997, which can be used by me for educational purposes. And we are talking, again, remember, we're talking of the process of fertilization. In this particular picture, the male and the female nuclei have not yet merged, so there's been entry of the sperm. It's like the entry of the gladiators, but only one gladiator goes in. The other 100,000 kind of wear the poor egg down, and then one goes in. And in fact, immediately that happens, there's a remarkable mechanism that stops any others going in, the blocked polyspermia, otherwise you'll have interesting triploid egg cells. That doesn't happen. And we come through these divisions which occur in nature in the fallopian tube until we get to this structure here. And this is the blastocyst, and it's going to be part of our discussion as to how do we think about this blastocyst. We're at about 7 days in a natural pregnancy, and what will happen is that if a pregnancy is, in a natural fertilization, if a pregnancy is to be established, and that happens in about 20% of the time, this inner cell mass will hatch through the outer layers and will form a little embryo that will cling to the wall of the uterus. And the outer cells here will form the supporting tissues like a placenta. So we've gone from here where we can't really detect any deterministic fate for the cells at all, to a situation here where at least some of the cells are going to become placenta and some are going to become the embryo proper. And once that has happened, we then begin to see the elongation embryo. We see a longitudinal axis coming. And after about 14 days, that's about 7 days after this stage, we just begin to see the signs of where, in 2 or 3 weeks' time, we'll see the central nervous system starting to develop. So these are the early stages. This little mass of cells, the inner cell mass, is going to be very vital to our discussion. So let's think a little bit about genes and development of that embryo. The first thing is the zygote. That's that first cell, the very first cell that forms from the merger of the sperm and the egg. And remember that 2 sets of genetic material don't actually completely merge until the second set of cell divisions. So they're still acting as almost independent nuclei in the first 2 cell divisions. The zygote must, by definition, have the genetic potential of everything, placenta, and the embryo that's going to form. And when we look at what happens is that of those 25 or 26,000 genes that the human possesses, we see an incredible array of gene switching going on. Gene switching which involves blocks of genes being switched on and off under the regulation of master genes. We have whole cascades of master genes. We have genes interacting with each other at particular times in particular ways. It's very hard to give an illustration of the complexity of this. But I was reminded of it when I was at a rock concert in Plymouth back in February, and I was standing behind the mixer desks, and the techies there. They had about 3 of these big boxes of resistors where they're sliding these up and down, and 3 computers to run the show for the rock band. And it looked very complicated, but my feeling is the array of gene switching that we get to form a complex mammal from an egg cell is going to be a bit more complicated still than that. And computer programs currently haven't managed to sort that out to, if you like, tell us what's happening. And so that's the early stages of this simply involve the regulation of the genes to do with growth of the cells. And it's not until this point here, the 4-cell stage, that the embryo's genes take over from the mother's genes. So up to this point, the whole thing is running on maternal genes, and then the embryo is now working with its own genes, and the developmental process can start. But let's now look at this mass of cells. This cell is totipotent. It will form these and these. This cell is what's going to form the mammalian body, the human body, and it therefore must have the potential to form every single type of cell that we possess. And depending who you talk to, the numbers you get for cell types in the human body range between 210 and 230, and I guess it depends how you define a cell type, how pernickety you are about your classification, what sort of taxonomist you are, splitter or a lump. But shall we say 210 plus cells, and the genetic potential to make all those lies in there, in the cell mass. So stem cells. I talked about stem cells in plants. Stem cells exist in animals, and we're going to think a little bit later about stem cells that we possess as fully fledged human beings. Let's think about them in general. They are going to be the source of many different types of cells, just as the stem cells I've worked with, shall we say, in a flowering apex will give rise to the petals and sepals and so on. The stem cells, a population of stem cells will give rise to many cell types. And it's been suggested that with the technology we now have available, and with the increase of degenerative diseases, as our Western population gets older, not dying off for other reasons, that these degenerative diseases have become targets for medical care, and we ought to be aiming to help people who've got degenerative diseases, or people who've been damaged by trauma or accident. And we might, because stem cells have the developmental potential to move in every direction, then maybe we can utilise that potential. And of course the blastocyst, that little hollow ball with the inner cell mass, as I said just now, by definition, is going to be, or is the structure that will, in nature, if pregnancy is established, give rise to all those types of cells. Blastocyst, the source of embryonic stem cells. There's a good deal of debate about how genuine a stem cell population this is. It's a bit transient. But nevertheless, whether we regard it as a fully fledged stem cell population or simply as a transient mass of cells that's got this potential, it's clearly got the potential. And so this is a little cartoon as to how, in general, a stem cell might work. In a real stem cell population, such as those that reside in our bone marrows, you have cell division will give rise to one cell that's going down a developmental pathway and one new stem cell, so the population is maintained. And then that developmental pathway, we might get that maintained, but eventually we start seeing what we call differentiation, that is the development of different types of cell. They're all there. So that's a sort of little cartoon. And then in relation to the blastocyst, this is how we might conceive of it working, that we might be able to culture these embryonic stem cells and then either in the body or in vitro persuade them to differentiate into particular types of cell for the repair of nervous tissue, muscle tissue, heart muscle, whatever it happens to be. That's the thinking behind the current debate, and indeed the current experimental work is going on in several places in the United Kingdom and elsewhere. So how actually will that turn into a medical therapy? Well, the first thing you've got to do is you've got to maintain the population of stem cells. Now, in my little cartoon I showed a couple of minutes ago, the cell division put one cell back in the stem cell pool and sent one down the pathway. And that is what actually happens in the populations of stem cells we have in our bodies as we speak now. But in the embryo, that population is very transient. They don't keep as stem cells for very long. So much so that Austin Smith, the stem cell expert here in Cambridge, says that embryonic stem cells are kind of like a figment of our nomenclature. That they may be totipotent or pluripotent, they may have this huge potential, but they don't exist as an ongoing stem cell population. Be that as it may, they're definitely there for a little while, they're definitely there for a few days. But they're not persistent, because they will form the embryo, and once the embryo starts to form, we begin to see, firstly, the formation of 3 different layers in the embryo: the ectoderm, the mesoderm, the endoderm. And then those 3 different layers, each are responsible for the generation of different tissues and organs. So it's a transient population of stem cells in the embryo. And this means that we need to identify the genes which keep them young. And it turns out to be 4 master genes. These are regulator genes, and those sort of genes I told you about that switch on other genes. And for those of you who speak the Celtic languages, will recognise Nanog as meaning of youth, which is true in Gaelic and in Breton and in Cornish, and I'm not quite sure about the Welsh. And it was discovered by Austin Smith, who was working that stage in Edinburgh and speaks Gaelic. So these 4 key regulator genes are absolutely vital for maintaining the youthful state, the stem cell state of these embryonic stem cells. You've got to keep them switched on. You've got to provide the circumstances in which they switch on. And this is going to come back later when we consider other possible sources of stem cells. And so what you then do is you persuade those cells to develop along particular lines of development in order to get a cell that might repair damaged heart muscle, in order to get a cell that might repair a break in the spinal column, in order to get a cell that might work on brain cells. You need to persuade them to differentiate. And of course, normally, this is a very complex process. It's like spatial patterning, as we call it, goes on in the embryo. Where a cell is important, as important as the genes that are switched on. And currently, there's a lot of debate about how we can actually induce this differentiation. So we're very experimental. And the technical hurdles to overcome are firstly that these embryonic stem cells are quite, if you like, fussy in their culture conditions, and only one lab so far has managed to grow them without having what's called a nurse culture, a culture of actively dividing cells from somewhere else that provides growth factors. And so you separate the 2 cells in a kind of, with a layer of material that's pervious to growth factors but not to cells. And so only one group has claimed the ability to grow these on their own. And so you've got mouse cells basically in the same dish, although they are separated by this layer that keeps the cells apart. But the second one may be even more difficult. One of my students, as part of her research, came down and interviewed Stephen Minger, who is one of the leaders in this area, a lovely man. And one of the questions she asked him was, is it possible yet to get particular cell types to differentiate to order? Can I come into your lab and order up some heart muscle, please? And his answer was no. Particular types of cell appear spontaneously in the culture, particularly the cells called fibroblasts, which contribute to our skin, for example. But in the main, you cannot get particular cell types. And the thinking currently is that we may have to inject embryonic stem cells into the tissue that is to be repaired, hoping that they will recognize that environment as part of the spatial patterning. And again, I'll say something more about that later. It might well be the location in the body that determines whether we can get the cells we need. So, embryonic stem cells, where 2 or 3 labs have been given licenses, and we'll hear a bit about more that in a minute. Where do they get them from? Well, the most common source of embryos for research both in the States and in Britain has been embryos that are produced by IVF. And as you'll know from early debates, the embryos, when embryos are produced by IVF, there are always spare ones unless the couple concerned insists that there are not, which can be done. And so you have these spare embryos, and the thought is we use them in research, and they can become sources of embryonic stem cells for our work. Regulated by the HFEA, again, anything to do with gametes or embryos outside the body, regulated by the HFEA.
Swallow down again, John.
The Human Fertilization Embryology Authority. And the initial round of licenses, King's College London, where Stephen Minger is the director of work, were granted a license, but Peter Andrews' group in Sheffield has got a license now, as have 1 or 2 others. But so that's one source of cells, but I wanted to consider another source of embryonic stem cells and go back to our idea of genes and development. The idea that the zygote, that first cell, is totipotent. It's got everything we need to make a fully fledged adult. And all the programming of the genes that I talked about. And if we look at ourselves, if we were to take almost any one of our cells, apart from the red blood cells, we would find that all our genes were there. So when a cell forms a brain cell or a liver cell, it's not that they lose genes. What they lose is the ability to express genes. The genes are there. The red blood cells are an exception because they lose their genetic material during their 2-week lifetime. And one of the big questions in mammalian biology, it was answered for plants and for fruit flies and for amphibians years ago, back in the 60s when I was a student here. They had already cloned frogs. They had managed to get frogs' genetic material to switch back on again. So an adult frog, the genes may be switched off, but they can be switched back on again. For mammals, that was a big question. Can we get them to start over? Can we get those genes to go back to as if they were in the embryo, even though they've been switched off for years, in many cases? And so what we're asking is can we restore that state? Can we in some way restore that state in which an embryo has now got, again, the potential to make all the cell types? And it's cloning. This is basically what we're doing, is we're cloning. We're trying to take genetic material from an adult cell and to generate an embryo with it. And of course there are two routes to clones. That all of us know clones, because we all know identical twins. And here we've got no kind of odd social situation. The clones are the same age, although, as I've always noted talking to identical twins, one always claims to be older. The one that came out first claims to be older. But I'd like to point out that genetically they're the same age, developmentally. Interestingly, cattle are twinned this way. The elite embryos are split, so that instead of having one elite bull calf, you'll have two or even four. But in human biology, human fertilization and embryology authority forbid us to do that. They forbid us to manipulate the embryo that way. So we can't start generating copies of particular genotypes. But of course, what I was just referring to about getting genes to start over again requires a different set of activities, and it requires this cloning, this nuclear transfer, taking the DNA that's been sitting there in an adult cell, doing the adult cell stuff, and getting it to start again in an embryonic situation. And here, of course, you'll make a clone, but the clone here is much younger than the donor. And in case of Dolly, that difference in age was 6 years. So Dolly was 6 years younger than her genetic mother. And this is how it's done, not to scale, but here's an egg cell with its one set of genetic material, and here's an adult cell, a somatic cell, as it's called, not to scale. If they were to scale, this cell would be a dot. And we take out the genetic material, or we irradiate with UV light to kill the genetic material, one way or the other, and then we transfer the genetic material from the adult cell. 2 copies, of course, because it's adult, and in it goes. So the 2 copies in the adult cell are replaced with 1 egg copy and 1 sperm copy. And by a lot of kind of manipulation, which may include an electric shock, very high voltage for a few microseconds, that cell is persuaded to become an embryonic cell again and starts dividing as if it was an embryo. But there's a lot of manipulation that has to go on. And the result of that first mammalian cloning is seen here. This is Dolly shortly after birth. I mean, she's quite a chunky looking lamb, but she's certainly a lamb. And this is the surrogate mother in whom she was born. You can see genetically different from the genetic donor. And here is Dolly in February 1997. It frightens me to think that it's over 10 years ago. It seems like yesterday in some respects. And she's giving a press conference there, and not saying very much, kept her thoughts to herself. At the time, kind of what I thought was an urban myth ran around the university and research sector that Dolly had been named after Dolly Parton. And I rejected that as just something humorous. But I was talking about 2 years later to Harry Griffin, who's the deputy director of the Roslin Institute. He said, no, it's true, we did name her after Dolly Parton, because the adult cells that donated the DNA were from the mammary epithelium, and I'll say no more than that. Apparently, Dolly Parton was told about this, and the sheep had been named after her, and she said, no publicity is bad publicity. I apologise for her joke, that's what she said. So let's go back to embryonic stem cells. If we were able to produce an embryo by cloning, what we'd have is an embryo that was genetically matched to the person who had donated the DNA. So a license, on that ground, a license has been granted to 2 labs to try to do this. Now, what's the point of this? Well, the point is that in many, many cases, when organs and tissues are transplanted between people, that it's very hard to get an immunological match, and so there's a lot of immunosuppression has to go on. In some cases, it still remains completely unsuccessful. Whereas if you were to take one of my cells and to grow a little mini-me in the lab, and I then needed a stem cell transplant, then those stem cells would be compatible with me and I wouldn't reject them. And that was the kind of pitch that was made particularly by the Newcastle Life Centre in applying for their licence, that a patient in need of regenerative medicine could wander into the clinic and a clone could be set up, embryonic stem cells could be generated, and they'd be walking out of the clinic a few months later completely cured with cells of their own origin. That was the kind of, very much the pitch made by the Newcastle Life Centre. And there is a kind of logic to it, that if we make embryos especially to fit the person. And it's I think two people have mentioned already, we've now got this very individualised medicine in the Western world. Firstly, it's my right to be repaired, and secondly, it's aimed specifically at me and at my genetics, just as people are talking about aiming pharmaceutical intervention, drugs and so on, at individuals' pharmacogenetic profile. So individualised medicine, and very, very technologically difficult. But there it is, licenses have been granted. And currently the situation is that using this route, that is of somatic nuclear transfer into a homologous, into a human egg, Newcastle generated one clone embryo, but it did not grow as far as the blastocyst stage. In Seoul, where a lot of kind of ethical fog surrounds what they were doing, we're now fairly sure that they did generate cloned human embryos by this route, maybe as many as 7. What they didn't do was to establish embryonic stem cell cultures from those embryos. And that was the paper that got into Science and was later rejected. And in a second paper, they claimed they'd made stem cell cultures from patients with degenerative diseases. That got into Science as well and was also rejected. But we're fairly sure that they did manage to establish about 7 cloned human embryos, which didn't go any further. So we can see that the technology is difficult. We can see that so far the aim of producing a stem cell culture, which remember is the baseline for this sort of therapeutics, the baseline is having the stem cell culture, has not yet been achieved with these cloned stem cells. Stem cell cultures have been achieved from IVF embryos quite readily. So here we are thinking about using human embryos, whether we generate them by cloning or whether we use spare embryos from IVF. And the question arises, and it's a sort of ethical question, that does our motivation or our mandate to heal allow the creation of embryos? It's the creation of embryos we're thinking of in cloning, or the deliberate use of spare embryos in embryos generated by IVF. Is that okay? Ethically, is that okay? Now, the former head of the Human Fertilisation and Embryology Authority, who is now head of the Charities Commission, former chair, I should say, is a close friend of mine. And we were discussing this over dinner one evening in London. And I just put that question to her. She's a Christian, and she said that compassion was one of the Christian virtues. And amongst other expressions of compassion, Jesus was a healer. And therefore she saw it as absolutely appropriate that we should use embryonic stem cells in a compassionate way as a method of healing. And that was definitely her take on the thing. And one or two others might want to come back to me on that. And you can't talk to her because she's not here. But that's the view that she took as a Christian working in this area. But others have brought other issues into the discussion. And try to get us to think about what it is to be human and how being human relates to the developmental stages represented by the human embryo. How do we relate one to the other? Philosophically, we can ask, what is a person, as Neil did yesterday? Well, these are just a few guidelines of what it is to be a human, just plucked out of the various levels at which we can look at human life. But here's a song that was written by Joni Mitchell for the Woodstock Festival back in 1969. Joni wrote it for a foursome called Crosby, Stills, Nash and Young, before Neil Young had left the group. And this is what the chorus said: "We are stardust, we are golden, we're a billion-year-old carbon." She must have known an astrophysicist, because she's dead right, that the only place is hot enough to make the elements bigger than helium are stars. And the only way that they can get out of the stars is if the stars explode, so we are all stardust. Intriguingly, the fourth line of the chorus says, "And we've got to get back into the garden." And I've always found that a very enigmatic line, considering what she's saying about our evolution. But we're a carbon-based life form, is what I'm saying. And then biologically, well, we're mammals. You know, that if you look at the number of mouse-specific genes and the number of human-specific genes we have, it's just a few hundred different. And people also make much of the idea that we're only 7 genes different from a chimpanzee. But I'd like to suggest that the number here makes very little difference to our discussion, because one gene can have a huge effect. If one gene affects the growth of the brain, then you only maybe need one gene different from a chimpanzee. But remember, those 7 genes have got 26,500 others to interact with. And the number of interactions possible between 26,500 is just enormous. And we just add a few more interactions to that. So the idea that we can trivially say that we're just apes with a few more genes is actually mistaken, because even one gene can make a huge, huge difference. And this is often used by people who want to trivialize the specialness of humans in the order of things. But there's more. We are the most complex of living organisms. We have self-consciousness. That means we're aware of us as separate entities from somebody else. We know who we are. Although sometimes it's difficult to remember. And I recall the name of the recent Bob Dylan movie, "I'm Not Here," or "He's Not There," or something like that. But we have self-consciousness. Now, one or two other creatures do have self-consciousness. They're aware of themselves as a different entity from others of the same species. It's almost certain, for example, that the porpoises and dolphins have self-consciousness. The great apes certainly have self-consciousness. Elephants have self-consciousness. And dogs perhaps have self-consciousness. Although one friend of mine who's a dog lover said, if his dog is self-aware, the dog, is not aware of that. Which I find sort of complicated. So there's lots of interesting philosophy going on there. But we also we have rational reasoning, intellect, language, moral reasoning, etc. We know the difference between right and wrong. And our moral reasoning is of a different order than the altruism that we genuinely see in social animals. Animals do have altruism, but our moral reasoning is very different from that. And what about spirituality? Is there another level of existence above that that's provided by our genes and the way they come together in our environment to form the human being? Well, yes, there is. And different religions express this in different ways, that we have another dimension, a spiritual dimension. We have a soul, some people would say. Some people would say that religious dimension places it along with nature if you're a pantheist. Others would say that we are part of the divine, again a kind of Eastern religion approach. The spark of God, which is a kind of theosophy approach, or the regular Christian approach is that we are made in the image of God with a spiritual dimension that is able to relate to our Creator. And that's very different from even the most advanced of the great apes and so on. So we are set apart like that, as beings with the God-given moral reasoning and the God-given ability to relate to the Creator. And so let's go back to the status of the early embryo, because this is where the debate has focused on whether it's appropriate to use embryos in research. And we're talking here about the moral status, whether they have moral significance, which is relevant to our discussion. And there is a range of views on this, and sometimes I get the impression that the different views are just talking past each other, because they're coming from completely different platforms to start with. I think Neil pointed that out yesterday. But here are two of the possible views. The early embryo is a human person. It has moral significance as a human person, as a philosopher would say. And often we see invoked in these discussions, we see the sanctity of life, meaning actually the sanctity of human life, that we need to treat the embryo as a person who should be accorded all the privileges of being as if he or she were already here. The sanctity of life is invoked by two Christian leaders in the West who have killed many people in an illegal war. I just raise that in passing. Sanctity of life is invoked in this argument by one of those leaders. And then it's not quite the other end of the spectrum, but that we cannot accord human personhood and therefore the moral significance that goes with that to an early embryo. Mary Warnock kind of stated it very clearly in her report back in 1990 that the committee concluded that they could not regard the early embryo morally as a human person, but nevertheless the embryo should be, and I use her words, accorded some degree of respect because of its special place in becoming, on its way to becoming a human. Now you can see that those two views may well have practical outworkings in what is legitimate for the way that we utilise human embryos. One of the things I've noted in the debate is that at least the media try to polarise the two positions as if the second one was the position that any reasonable, rational person was bound to reach. And the first one was an old-fashioned religious view. I was talking to Evan Harris about this, who was the Liberal Democrat spokesman on health and science affairs. I mean, he's a very caring man in terms of the disadvantage in society, but very intolerant of anything religious. And he just dismissed the first view as the viewpoint of a load of fundamentalist Luddites. I'm quoting exactly what he said. So people who hold that position are being, if you like, demonised. The Independent recently had a picture of an early embryo, the 8-cell stage, and it had above, is this a ball of cells or is this a human being? So they were polarising the debate too. I'm sure that we need to think about how we're going to ask, are we asking the right questions? And if so, if we are asking the right questions, how are we going to answer them? Now, obviously our worldview may affect our views, but when we look at the views that are held, and I'll take those two, if you like, as two different points of the spectrum of views. When we look at the way that people hold those views, it isn't actually religious people versus non-religious people, because there are significant numbers of Christians, even within the Reformed Protestant tradition, who are happy to accept the use of embryos in stem cell research. They are okay about it. And others who are not. The Mori poll conducted in 2001 with 2,000 adults had something like 75% of the British population supporting embryonic stem cell research, 18% against, and the rest didn't know. So that 75% certainly included some people of Christian persuasion, and the 18% certainly included people who had no religious belief at all. So it doesn't break down completely on religious grounds. And then we have this way of interpreting Psalm 139, which I always like to point out was written at a time when pregnancy was understood in the way that I delineated at the start of the talk. I seem to have got a jam here. He didn't want to move on. So if we look at different groups of Christians, Jewish believers are very positive about these developments, and in fact, the Association of Jewish Rabbis, going from liberal to orthodox, have severely criticised the Roman Catholic Archbishop of Glasgow for raising objections to stem cell research. In Islam, we have this mix of views. And we've also got this situation that has been raised both by Neil and Tim as to how we deal with this tension between our own morality and if society reaches a different conclusion. And Michael, are you going to be sort of talking about that in more detail? How do we handle that?
I'll prepare something.
Oh, right, good. I'm glad it's going to be spontaneous. But currently, for example, the Lawyers Christian Fellowship is mounting a legal challenge against the Human Fertilisation and Embryology Authority for granting the licences for hybrid embryos, which we'll come to in a moment, on the grounds that they've acted illegally. And so they're trying to, if you like, to use a legal route to raise the awareness of their position in a community, a society which has accepted another position. How do we cope with that when society is going down one track and our morality doesn't like that? We don't, I hope, go around bombing clinics like happens in the United States with abortion. Now, interestingly, views across the world differ on this. We hear a lot about embryonic stem cell research being banned in the United States. It's not true. Embryonic stem cell research is banned in the United States only in labs and clinics that are funded by the government. That anybody else can do as much research in embryonic stem cells as they like, and indeed a lot is going on. If you Google embryonic stem cells USA, you'll find loads and loads of drug companies, biotech companies, and charity-funded labs are working in embryonic stem cells. Italy is the most interesting country that until recently, almost any research in embryos was complete free-for-all, and hence the couple who went to Italy to try to have sex selection for their embryos. Embryonic stem cell research was allowed, fertility treatment for women well past the childbearing age was allowed, and so on. The grounds being that, well, surely, a good Catholic country like this, there'll be self-imposed limits, but there weren't. And so they've now gone from being completely free to completely draconian in their regulation, and it's now completely illegal to do research in stem cells. And this is a picture of Stephen Minger. You can see he's a bit of a Californian dude here. And the poster talks about how if he was caught, he'd be fined €150,000 and be subject to up to 6 years imprisonment, whereas in Britain he's allowed to freely pursue this practice. And Stephen gave that poster to my student when she went to interview him. Now, where are we at? Is this technology as promising as has been claimed? Or is there a lot of hype here? And Lord Winston, I think, hit that question on the head very, very clearly in his presidential address to the British Association back in 2005. His view was that it was all hyped, that in order to get funding, the medics and scientists had exaggerated their case. And parliamentarians were led to believe that clinical treatment was just around the corner. Now, depending on what parliamentarian you speak to, some say, yeah, I think we were deceived, we were duped. Others are not so sure. They think that they were fully aware of the limitations of the technology. And indeed, if you look at Hansard, even Evan Harris was cautious in his espousal of the technology. But nevertheless, Lord Winston was quite suspicious of the optimism. And this was in 2005. So this was some 4 years after the change in the law that allowed us to pursue this research. So 4 years down the line, and it's now 7 years down the line. Do we remain optimistic or pessimistic? This embryonic stem cell research will get us anywhere. I think I need to say that despite the hype, that at this point, no new therapies have been developed with human embryonic stem cells. Some 7 years down the line from the change in the Act. And none are indeed on the cards. No clinical trials have even been initiated. So that might affect our thinking. Let's then, for a few minutes, go to the adult human body and think about the embryonic, the stem cells that are not embryonic. And once an embryo starts to form into an actual little fetus, then stem cells begin to become located in particular places in the body, and they persist throughout life. Located at special sites, the bone marrow are the ones you all know about, and even as we speak, the bone marrow is dividing away, producing all the different sorts of cells of blood, and we're producing something like 8 million red blood cells a second, because the blood cells turn over very fast. And I always like to think, you could put a microphone to your long bones and hear them dividing. Speaking as a cell division person, I like to think of that, as they grow apart. And of course, their function is to in the case of the blood system, our cells are constantly turning over, our skin cells are turning over. Sometimes we need to repair stuff when we get wounded. When I tear a muscle and I'm out running faster than an old man should be running, they repair at ever-decreasing rates as I get older. But that's the sort of thing. That's what they're for. And in nature, of course, their potential is limited to the job they've got to do. Blood cells, muscle, whatever. And the niche is very important as well. That part of the stem cell behavior is based on the location in the body that you find them in. That's called the stem cell niche. Now, when we think about their use in therapy, in regenerative medicine, you would automatically think that because their role in nature is relatively limited, and their role in therapy may be relatively limited. But bone marrow transplants have been used to replace bone marrow, and that's clearly, that therapy is very well established, and I suspect that the 70 therapies that are talked about by some of the Christian groups include what I call regular bone marrow to bone marrow transplantation. And that works. But is it possible that we might persuade them to do something else? Could we take a stem cell that, shall we say, was bone marrow, and put it in another niche, another environment? And would that environment persuade it to differentiate into something else? We call it trans-across differentiation. Is that ever possible to do that? And indeed it turns out that at least for bone marrow cells, and maybe for one or two other adult stem cell types, trans-differentiation is a reality. Now, the clinical trials have been a little bit fogged because the controls have not been brilliant. But I think my view of reading all the literature is that the clinical trials that are going on do look as if bone marrow stem cells can trans-differentiate in a new niche to form things like heart muscle. So adult stem cells, at least bone marrow adult stem cells, seem to have this potential. Where you put them, the niche seems to be important. And then we've got other indications that there are other sources of stem cells. The cells that form the sperm, just before they divide to take me down from 2 sets of genetic material to 1, they have a stem cell-like character. And culturing them, this has been done with mice in a lab in Tübingen. And there we go. They have a stem cell-like character, and they can be persuaded to act like embryonic stem cells. And will behave, if you put them in a mouse embryo, they behave just like embryonic stem cells. So here's another case, and the professor of anatomy in Sheffield glibly said it would only take about 150 biopsies to have a range of immunological types that satisfy most people's immunological needs. And then he also added, the biopsy isn't that painful. I've crossed my legs by this time. And then the cells in the cord, when a saviour sibling, that is a sibling who's been generated by genetic selection in IVF, when a saviour sibling is born, they always hope they don't have to tackle the baby's bone marrow, but get the cells from the cord, and that's generally what happens. But cord cells have also been persuaded to trans-differentiate into liver. Tiny little liver-like organs grown in Newcastle. And then we've had more. The bone marrow cells have been used on a kind of former to grow a heart valve. And then even the Guardian isn't immune from the breakthrough type hype headline. So these were bone marrow stem cells that were persuaded to go down the developmental route as if they were spermatogonia, sperm-generating cells. So all sorts of things are happening that indicate that adult stem cells and other similar sources do have some potential. Blood vessels grown from bone marrow cells. And one of the questions that we're raising here is it legitimate, particularly for people who are ethically sensitive about the use of embryos, is it legitimate to now push instead the strong use of adult stem cells for the sake of those whose conscience is concerned about embryonic stem cells? And there's a lot of, I think now what's happening is the opponents of embryonic stem cells are now hyping adult stem cell research just to make their case stronger. And you've got to kind of steer your path very carefully between the hype from one side and the hype from the other. Now, there are other dilemmas which I want to deal with very quickly, and one of which will be the hybrid embryos, since it's right up to date. But here they are. And it's a very interesting commentary on the way we deal with these issues. These applications were made at the end of 2006, and the HFA on January the 11th, 2007 said, not for now, thank you, we want to pause. I remember the day very well because I was giving a Christians in Science Christian Medical Fellowship lecture in Southampton, and I needed the data of what the HFA had said. And they hadn't made the decision by the time I got on the train, so I had to log on when I got to Southampton. But they said no, and they wanted several months to talk about it. No, how did that get there? Yes, let me just. I'm just going to move that. Excuse me. That shouldn't be where it is. Oh no, maybe I'll leave it there. I will leave it there. I was going to leave it while we actually talked about what's being done, but this is effectively what people are asking to do. They're asking, instead of using a human egg as the receptor for the adult DNA, to use the egg of another mammal, and this time I put in another part of the cell. This is the part of the cell called the mitochondrion. It's the part of the cell that generates the energy for cellular life. And I put it in there because it contains a tiny handful of genes. It contains about 20, alongside the 26,500 that the nucleus contains. But unlike the nucleus, there are multiple copies. There are several mitochondria in the cell. In fact, in some cells, hundreds. And so what will happen is that if we take the genetic material out of the rabbit egg or out of the cow egg, and those are the two mammals that have been proposed, and we put in the cell from the adult human, the DNA from the adult human, then you get 26,500 genes from the adult human, but 20 from the rabbit, or 20 from the cow, but those 20 being multiplied. So we left the mitochondria there, and that may affect some people's thinking about this technology. Okay, let's move on. Now let's go back to the political world. The HFA had said no. They said no in January. But the Parliamentary Committee on Science and Technology said, you can't do that, I'm sorry. This research comes under the aegis of the 2001 Act. So you jolly well got to let them do it. And so they were given the go-ahead just 5 weeks later. And the Newcastle Life Centre and Edinburgh were given the go-ahead. But we're in a very confused situation here because other parliamentarians have said, well, sorry, this didn't come under the Act, and we therefore need to debate it. And so the 2007 bill, which is now the 2008 bill, now includes the clause to put in these so-called hybrid embryos. Our political world is very confused. The Parliamentary Committee on Science and Technology pushed the HFA into saying yes, and this has happened before the bill has become law. But in 2008, the Newcastle Life Centre reported the formation of one, what should we say, hybrid embryo. I don't like the word hybrid because these are not real hybrids. One hybrid embryo under the licence that they'd been granted in May 2007. So we've got a very confused political situation, but the point I'm trying to make here is that pressure from those who had an interest in getting this research going pushed a semi-independent body, the HFEA, into making a decision before they were ready to do so. And apparently before Parliament had eventually given the go-ahead. Very confused. And then there's a tiny bit more to think about. I said earlier that there'd been no therapies developed from embryonic stem cells, where there have been maybe half a dozen developed from this new use of adult stem cells. It's certainly not very many, but there are a few. But one of the reasons that nobody's going ahead with clinical trials with embryonic stem cells is because of this presence of the mouse cells in the culture. And no health authority is prepared to risk the possibility of litigation if some harm comes to somebody undergoing clinical trials, not because of the technology itself, but because of the passage, for example, of oncogenes, viruses, and so on, from the mouse cells to the human cells. That seems to be the hold up. So we haven't yet got to a position where anybody is even willing to allow a clinical trial with the embryonic stem cells. And we'll just have a few more, just a couple more minutes, Dennis, if I may, to talk about this very recent development. The stem cells in the skin, which repair our skin when we get injured, are called fibroblasts. And several groups have managed to turn them into embryonic style stem cells. They had to be induced pluripotent stem cells from fibroblasts in the skin. And in order to get them to do that, you've got to switch on those 4 master genes that I talked about. You can't switch them on. It turns out that they won't be switched on. And so what people have done instead is they've genetically modified these cells by putting in extra copies of the 4 genes you need for them to behave like embryonic stem cells. You sort of get that the 4 master genes are switched off and won't be switched on again. Sorry, we're fibroblast made, we're not going to be embryos. And so the genes have been put in an active state, they're genetically modified. And some people hail this as a great ethical breakthrough, certainly all over the States. The conservative Christian press hailed this as an ethical breakthrough. And then, initially this was done with mice. The 2 papers that appeared in generated discussion were done with mice. But just this week, we've now got embryonic stem cells produced by that method, by the iPSC method, from the skin of patients suffering from degenerative diseases. And so maybe we're seeing what I might call a breakthrough here, that people who need repairing, regenerating cells in them, the issue though remains, what about the 4 genes that have been put in an active state? What effect will that have on the future use of those cells in therapy? So a little bit of a plug. There are 2 books. Introduction to Bioethics is nothing as deep and as thoughtful as the sort of book that Neil has written, but my students find it readable. This website here, this is the website of a project which Michael Rice and I were involved in as advisors at the University of Bristol. Bioethics Education Project, ACUK. And although we set it up for students in year 12 and 13, that's the last 2 years of high school in Britain, it's been very useful for teachers and so on. And then this resource here, this is the Bioscience Centre, the Higher Education Academy, has got a number of ethical briefings, and Chris Wilmot and I are currently writing 2 more which deal with some of these issues. But I think the final comment I make is you can see that very much, this is very much up in the air. And the debate has been conducted very much under utilitarian lines. And as Neil said yesterday, if you say that you don't want to do this work, then you're facing down these patient groups who are lining up behind them, these tens of thousands of people suffering from degenerative disease. And the debate often gets quite heated. So much so, for example, Christopher Reeve, who played the Superman in the 1978 series, the 1970 series of Superman films, was a great supporter of embryonic stem cell research. And in one particular discussion, in one of the American more serious chat shows, said to the host, bigots are delaying my recovery. So there you are. If a position you want to take is that you're opposed to embryonic stem cell research, and you're facing down the people who are supporting it, they will call you a bigot, which seems to me not a very good way of conducting the debate. We are speaking past each other, we are taking different positions. And again, I quite deliberately have not given my own position on this. I'll try to conceal it through the debate, but I probably won't be able to. Thank you, Chairman.
Thank you very much, John. Right, good. So we've got some good time for questions and discussion. Yes.
Hi.
Just really rising from your very last point about Christopher Reeve, one possible response to that, of course, is, and I'd be interested to know what your opinion is on this, is the allocation of resources into the most effective therapies. So it could be argued that if we have got a lot of therapies already with increasing numbers from adult stem cells, is that going to be a wiser use of investment in bringing in something that hasn't as yet produced something? So that's my first question. Secondly, you said partway through the talk about the big impact that one gene can have on development and the importance of that. And that raises questions in my mind, which I'm interested in comments on the hybrid admix communities and the impact, therefore, that those 20 or so mitochondria genes had on the development. And it's not, I mean, obviously you mentioned the fact that it raises ethical issues, which it does, but does it really raise practical issues of how useful it is to have this hybrid embryo for? Is it going to ever lead to therapies, or is it purely just a research tool that they're creating?
Okay, so the first one was about allocation resources. Now, I need to say that the new therapies that are being developed from trans-differentiated adult stem cells, and I do make that difference between the regular stem cell transplants that are built in now to, for example, treating some forms of leukemia. Those therapies are not new. The new therapies based on getting adult stem cells to change their pattern of differentiation are not in clinical use. They are under clinical trial, and we don't yet know whether they will go into clinical use. And they're based on trials of repairing heart muscle with bone marrow cells. But it is the most hopeful we've got so far. My own personal view is that actually adult stem cells have been ignored in the debate. Whatever I think about embryonic stem cells, adult stem cells have been ignored in the public debate. And I think that we ought to be allocating resources to therapies that do look hopeful. And that may well be with adult stem cells. And I think that they maybe ought to have more publicity in this. They need an agent, really, don't they?
Well, in that, I mean, just quick sort of that, because cord blood is getting a bit more.
Yeah, well,
with banks, banking it.
Indeed. But cord blood, in most cases, will behave like bone marrow. But it does have some embryonic features about it. The other one was about the mitochondrial genes. When you look at the sequence of the mitochondrial genes in rabbit and in cow and in human, I mean, the 20 genes have exactly the same function as those 20 genes in human mitochondria. You can't distinguish between the functions. And if you do an admix in the lab between the product of the mitochondrial gene in a mouse and the mitochondrial gene in a human, they mix completely well. I need to explain that technically the mitochondrion needs a huge contribution from the main genome, and some of the components of the mitochondrion are built with a bit that's coded for, controlled by the mitochondrion, and a bit that's coded by the nucleus. They have to join together in structure. And when you do the admixtures between mouse and human work completely well. The proteins, the structures are the same. The sequences are very similar. You know, the sequence variation between the two is fine. So I wouldn't be, I personally am not worried that the presence of the foreign mitochondria will make a difference, if you like, to the development of the cells. But nevertheless, I still would be a bit more concerned about the cytoplasm, the cell contents. An egg is a big cell, it's got a lot of stuff in it, maternal influence lasts quite some time. Will there be any carryover material, such as a latent virus, that might affect? So I'd be only inclined, let's say I was supporting this, I'd be only inclined to support it for research purposes only, to show us how we can grow stem cells in clone embryos.
Can I just add a little point? I mean, I personally, I think I'd be very cautious about allowing the hybrids to be used therapeutically, unless there's a huge amount of research actually on their function. I mean, if you think about the consequences, if you're trying to heal heart muscle or something which has to have a certain level, the mitochondria are pretty important, in the energy metabolism of your cells, and for heart muscle that would be critical. And something even if it would be a very subtle underperformance or overperformance of energy metabolism in the cell, it could have, I think, serious consequences. So although I agree with John's point in the lab, things could look very similar. I think one would have to be very, very cautious before wanting to use hybrids for therapy rather than research.
I'd only want to see these research.
Yeah.
Without a doubt.
Yes. Yes.
Have they managed to overcome the problem of telomeres with the production of pluripotent stem cells from skin fibroblasts?
They haven't been, telomeres are the structures on the ends of chromosomes, which are copied in a very special way to stop your chromosomes fraying at the ends. But actually, over our life, these do shorten. And so you can almost tell the kind of life of the cell by how long this telomere structure is. And we don't know yet whether or not the induced pluripotent stem cell state will restore the telomeres to embryonic length. There's a very big debate about this, exactly how much of DNA damage is repaired when you do somatic cell nuclear transfer. Does the embryonic status confer high-level repair activity? With Dolly, it may not have done. So the telomeres are a special example of that, and we just do not know yet. They haven't been around for long enough.
Yes, questions?
Shocked silence. That's good.
Yes. Yes.
I think there was did you say with the skin stem cells that you could place them in an embryo and get them to behave
Well,
as if they were embryonic?
now that was the stem cells that had been generated from the mouse testis.
Oh right, yeah. Well, but I mean in that case, if you've got stem cells that are behaving like embryonic cells, then what's the ethical difference between embryonic stem cells and stem cells that behave like embryonic stem cells? If it's all dependent on their potential.
I think that is a really intriguing question. I raised the question with a discussant from the Christian Institute on Premier Radio. Exactly what are you conferring your idea of the sanctity of life? And you're generating embryonic type stem cells without generating an embryo. And so are you saying that these should be afforded, if you believe, that is, that the early embryo should be afforded the full status as personhood, are you saying that this group of cells that you've made by a different method, whether it be by somatic cell nuclear transfer or whether it be by transformation of spermatogonia, are you now conferring on those the status of personhood?
Exactly.
So I leave you to think about that.
But can those particular cells, though, form a new individual? Isn't that the difference between an embryo and something else?
What we don't know, Gareth, is if, let's just say that we took all the inner cell mass from the blastocyst and put these spermatogonia-derived stem cells in their place, whether they would make a mouse. We don't know that. What we do know is if we put in tagged spermatogonia-derived embryonic stem cells, they behave amongst other ES cells as if they were ES cells themselves, and we can follow the tagging through the developmental stages. But it would have been fascinating had the group in Tübingen removed all the inner cell mass and replaced it. And the answer is we just don't know. But what sort of ethical status would you accord to that embryo if you'd done it that way? It's not certainly not a unique human genotype. Fascinating. That's a fascinating question. Let's go in. Think about that.
Sorry for non-biologists here and people who might have got a bit lost on the distinction between all these various things. If you feel a bit lost,
if I'm being too technical, please
come up with questions, because
ask.
sometimes it is there are quite subtle differences between some of these things that. So don't hesitate to ask if something's got. Yeah, so go. Yeah.
Can I ask you the general question, John? I mean, do you think that in this area there is such a thing as ethical research and non-ethical research? Because clearly this is a distinction which is made by some. And in that context, would you like to comment on altered nuclear transfer, or the ANT technique, so-called, that of course a member of Bush's Bioethics Council has certainly advocated as being ethical research?
Yes, now I thought the altered nuclear transfer was a con, actually, because it seemed to me that...
Can you explain the technique?
Yeah, can you remind me the details? I remember reading, thinking that. And I know that Nigel Cameron made a very big play about this when I spoke to him, but I can't remember the details.
I think you simply again switch off some genes so that instead of developing into an embryo, in a sense you're developing an abnormal embryo,
That's right.
which cannot become and individual.
That's right. So what you're doing, instead of allowing the later divisions of the early embryo, you're stopping it at an early stage. But if you want to accord human personhood to the one-cell embryo, then you're still in trouble, because what you've done is you've stopped it developing into a blastocyst and then into a fetus. So I felt that was, I think it had been misunderstood what was going on there.
There was you can watch a whole debate on that particular one online, actually. We had a whole discussion on that in the Faraday lectures about a year ago, I think. So, Sir Bryant and someone came over from the States to discuss that one. So, any other questions? So, yes.
Can I just ask, sort of looking to the future then, are you prepared? You haven't really looked ahead. Are we going to see a move away from cloned embryos as a source of stem cells? Is that what you're saying is going to happen?
I don't know. The pressure to continue the work, whatever you think about it, I think one has to acknowledge, whether you're a supporter of the work or not, that the publicity and the pressure for Britain to carry on with this work has been enormous. And it's been done by lobbying, it's been done by presentations in the press. Back in the 2001 modification to the Act, the Human Fibrosis Act was being debated. Ian Gibson, who was then chair of the Parliamentary Committee on Science and Technology, was talking to the leaders of many of the learned societies. I was then vice president of the Society for Experimental Biology, president-elect, and he came to talk to me about it. I knew him anyway because he was a former lecturer in molecular biology. And one of the things he said to me, John, he said, you need to make sure that your friend Susie Leather, who is the chair of the authority, is up to speed on this, because we want this to go through the HFEA. His words were, Britain's got a lead in this area, and I heard him make a speech in the House of Lords to the Lordships. Britain's got a lead in this area, and it must be maintained. We've got to stay ahead of the game. So there was a huge amount of political pressure for embryonic stem cells, and I think that will continue, because it has its very, very influential supporters in the Commons and in the Lords. But I think, for me at the moment, and this is, I'm not speaking here, if you like, ethically, if I can distance myself from that, I probably can't, but I'm not speaking ethically, but for me, just standing here as an interested observer with a good deal of scientific knowledge, it looks to me at the moment as if these other forms of therapy with stem cells are nearer to producing a result, as we would say in football. That we are nearer to something that can be used in the clinic than we are with ES cells. So that's my view of the state of development. And talking about ethics reminded me about the Gareth's other question: is there ethical or non-ethical research here? My view of the scientific enterprise is that, and speaking as a Christian, that God does allow us to investigate the universe in which He's put us and I don't think any area of knowledge itself is off limits, but there are unethical ways of obtaining knowledge. A classic example would be that we don't force human breeding. We just wait to see who mates with who to get our genetic pedigrees, for example. So, is there anything unethical about any of the research going on stem cells? Well, that is almost certainly a matter of opinion, as within this community of Christian believers or Jewish believers or Islamic believers, people try to work out what it means, for example, to be a follower of Jesus and think about these issues in the light of scientific knowledge and the light of their development of their virtue ethics. Different people will arrive at different conclusions as to what is ethical and unethical. Some people believe deeply that embryonic stem cell research is deeply unethical, and others say, no, that's fine, that's one of the tools that we are permitted to use.
John, to follow up on that, this morning, in the context of genetics research and so on, and genetic testing, you were mentioning how you would yourself come around to the thought that a slippery slope actually was a real danger. Having been perhaps a non-slippery slope man, you were kind of seeing more, slippery slope really does seem to be
Yeah, I'm
happening
seeing
in
the
that
black
area.
runs, yes.
So I wondered if you could see, looking, down in the future, I mean, can you see that you might begin to feel the same way in the stem cell area? Do you think there are areas in that area of research and its applications which could go the same way as genetic testing, in that sense that right now perhaps it's not an obvious slippery slope, but there might be one waiting in the wings for us? What do you think?
Now, slippery slope waiting in the wings is quite an analogy. That's a beautiful metaphor. Yes, yes. I don't know. I mean, some would say that it was an ethical precipice, not a slope at all, and we stepped over it when we started to do work on embryonic stem cells. Not a position I hold, but I can't see. I mean, I suppose the slippery slope might be that we might be tempted to use more and more experimental therapies, which were not tried and tested, on vulnerable patients who were very anxious to have a cure for their condition. And this also relates to some of the stuff that Tim was saying, that that death is regarded as always regarded as a moral bad, not a moral good. So there may be patients who are very vulnerable and who are persuaded by enthusiastic biomedical scientists and clinicians that this is going to sort you out some. Let's have some of these. There was a drama doc on Channel 4 which included Susie Leather and Stephen Mingo, which raised just that scenario that experimental treatments would be offered when they were not yet really checked out. But that's only a tiny slope, I think. I can't think of a long one that's going to take us down several thousand meters.
Let's just to follow up one question. I mean, that's how as an evolutionary biologist, I'm really interested in, these missing links we have between us and, these other primates and so on. So I take a chimpanzee embryo and I take some human stem cells and I make a chimeric embryo, and I wanted to see develop, because I mean that would be really, really useful, wouldn't it, to see.
Okay, well that might be a slippery slope. Yeah.
Yeah. So what do you think?
Yes. No. I think having moved the area of consideration slightly, and incidentally, chimeric embryos are also under the bill that's being debated right now. And I have considerable misgivings about them. I will say what I think then. But yes, you're quite right. There is a slippery slope there. What happens if some gung-ho group decide, well, let's implant one of these embryos into this willing recipient to whom we paid $25,000 to bear a chimeric embryo? Yeah, that could be a slippery slope. And I think I'm beginning to feel quite uncomfortable here.
So I believe that's in the present act.
Chimeric embryos are in the present act, and
yes.
I wonder how on earth they ever got in there.
Yes.
Because I myself, speaking as a developmental biologist, cannot see any justification for them. I mean, it's like I was just saying to Gareth, there are areas of knowledge which I think are not out of bounds, but the methods we have of getting there are out of bounds, and that to me would be an out of bounds thing. It just, why I would ask the question, why do we need to do that? Philip has got a waving her arm.
Well,
okay,
yes.
Really quickly on at what point in the development you're accepting in many ways the size of the embryo, which has just a huge input, we don't know exactly from an animal. But at what point you say this is no longer a human and becomes an animal? At what point you're not happy with 50-50 potential hybrid definition at the moment? But at what point?
It's very clear. It's very clear to some genetic manipulation experiments over the years that putting a couple of human genes into a sheep does not in any way make it a human. Its telos is still sheepy, and because the mitochondrial genes function exactly the same way, and I take on board Dennis's concerns about subtle differences. It may be that we put some human mitochondria in there as well, depending how we do the transfer. I don't regard in any way as that entity that develops as being in any way subhuman. I absolutely don't. Otherwise, you'd have to say that people who have had a transplant of a pig heart in some way were now pigs. because I mean that's actually a functioning adult being who's got a big cell and a monkey cell. But at what point do I say? Well, let me be very honest, I don't know. But the 50-50 seems to me to be an abomination. I'll be quite strong on that.
There's a question at the front, yes.
Probably a very naive question, but at what stage in embryonic development, and especially in chimeric embryonic development, are you talking about humanity, a human person?
Yeah, I mean this is a nice rephrasing of the old question about when does human life begin. And I always point out that question, phrased that way, doesn't have any meaning, because the sperm is human life and the egg is human life and so is the zygote. But it is, when is it a human person? And I think the question is impossible to answer. My own position, I will start to play my cards a bit, is that the few-celled embryo is not a human person. But at what point we decide that we are dealing with a human person along the scale of development from the implantation to forming pregnancy through to the birth of the little one, I do not know. But I think it is a gradualist thing. And whereabouts in that gradual development you think that you should no longer treat this as being of no worth, I do not know. But I am fairly sure in my mind that 24 weeks abortions are too late. And I know that John Wyeth's data, and he's not the only one to achieve such a high success rate with 23-week embryos. If you go down to 20 weeks, then your success rate virtually reaches nothing. And that's nothing to do with technology. That's to do with the development of the brain. It's just not able to sustain any further development outside the nurturing mother. So, should you take that as a point where, the brain is now developed enough for, with given medical intervention, the baby becomes viable? Is that a point you'd want to talk about as being worthy of consideration as a human person?
I'm asking you.
No, well, I'm throwing the question back because I think everybody here will have a different viewpoint in the developmental stages from the start of pregnancy, which is a very significant event, only 20% of them get that far. To the birth, I think somewhere along that line, each of us has got the point where we say, this is now a morally considerable, as the philosophers say, a morally considerable being. So, you make up your mind, I'll make up mine. But I'm certainly unhappy with late abortions. And I think that's because I'm beginning to see that growing fetus as a human being.
I think that would be a really interesting question to bring back to the panel tonight. And
thank you.
It would be good to hear all the different views. Okay, I think we'll make this the last one before tea break.
Yeah, it's a heavy burden to bear there. First, just very briefly, I thought I'd give a supposed answer to your question. You say, why would anyone want to do it? Because we can.
Because we can, yeah.
Yeah, right. And
I can go and shoot an old lady on Modern Bridge, but I don't know if I should do it.
Well, Freeman Dyson has commented in a film, in a very well-known documentary about the atomic bomb, about the technical arrogance of science. He talks about being able to move 100 million pounds of rock into the air, this kind of thing, and what the feeling that it gives you. We can do this, and it clearly did run the thinking of some people in the Manhattan Project for a while. We can do it, so let's just do it. And a similar concern could be given. But relative to this last question, I think the most, in my view, as a historian, it's more of a disciplinary view, in my view as an historian of science and Christianity, the most fundamental question about science and religion through the historical periods and all these issues is the one that was just asked. And it has to do with what I would call the problem of M&Ms. You have in the 17th century with the adoption of the mechanical philosophy, you have matter and motion being the explanatory terms for the universe. And then you have these other M categories like meanings and morals and minds. And nobody, flat nobody, has any idea of really how to put those together and make sense out of that. But it's really the same question, I believe, in another form, the same question that's come up. Because, of course, there are people like John Searle, your co-author apparently on this introduction to bioethics, who thinks that those minds don't exist.
Another John Searle, not the same.
Oh, okay. All right, I take that part back. Same spelling. Okay, there's a famous
Same
philosopher.
spelling, same name, yeah.
Same philosopher. First
Yeah,
famous philosopher of mind.
I think he stole my book.
Well, you know, he doesn't believe that minds really exist
I know.
in that sense. I mean, and a lot of serious philosophers don't believe that mind exists, and I don't think we should need, I don't know how you talk to someone who's of no mind. I mean, really, but that's the case. You can see the point I'm making.
We say talk to the hand.
Yeah. And so, you know, and this drives the origins controversy, in my view. It really does. It drives the origins controversy, both in the States and elsewhere. Is this the fact that nobody really knows how to put those two things together? And so you have people who focus on minds, on meaning, rather matter and motion, who don't think these other things have any reality at all. And then you have people for whom those things have great reality, the fundamental reality. And these other ways of describing things are greatly limited in their ability to do things. And it seems to me that that's the issue that we need to get at more rigorously. So maybe it's all up to the philosophers in the end.
Yeah, I mean, I think I will leave you with that comment, and I think it's very interesting, and achieving a synthesis, I think it could be a major project. But I'll come back to your first comment. It's absolutely true that scientists as a community are no more or no less prone to hubris than anybody else. And I have certainly met and talked to scientists who would be quite happy to reproductively clone another human being just for the hell of it, if I may use that expression. And so if you've got one or two of those guys about, and they've got the facilities, and I said, well, where are you going to do this? And he said, well, Mexico City is as good a place as any. Got the facilities, and I can bring the bugs. You know, if they're going to act outside the law of their own country, I'm not quite sure what we can do to stop it. So it may well be that hubris and arrogance on behalf of a very small number of people wins through in the end for them. So it's quite a sobering thought, isn't it?
I think it'd be great to bring, again, all these things back to the panel as well this evening. Hubris, arrogance, and, you know, who is a person, and the other things, to keep the panel really working hard. So please do put your... this box is labelled Cambridge University Press, so don't be deceived. This is actually the questions box. And
they're going to be published tomorrow.
Yeah, they won't be published. But just pop the lid off, and there's also some pieces of paper here, so I hope you'll use the tea break to start really getting those questions down. And again, I think we worked John really hard. So I think we want to thank him very much. So.